Okay, we're going to look at chromosomes and DNA replication. This is going to explain how the genetic code is contained within our DNA. When we're looking at the DNA and the chromosomes, there's two types of cells. We have prokaryotic cells. These are single cells. They have circular chromosomes. This is representing the DNA, the chromosome material, just within the cell. And then we have eukaryotic cells, which contain a nucleus, and then within the nucleus, we would find our DNA and chromosomes. The chromosome number varies between different species, in other words, between different organisms. Humans have 46 chromosomes. You guys need to know that. Mark it, highlight it, something. You need to know that humans contain 46 chromosomes. The giant sequoia tree contains 22 chromosomes. And then our little fly down here, he contains 8 chromosomes. So every species has a different number of chromosomes. When we look at the length of DNA, DNA is more than a meter in length, but it's still within the nucleus of a human cell. And so we wonder, how is that possible? Well, the DNA is going to coil and wrap around proteins tighter and tighter and tighter until it fits inside the nucleus. It's the chromatin and chromosomes that we actually find inside the nucleus, and we'll take a look at that. When we take a look at the chromosome structure, let me get my pen. Okay, this is representing the cell, and then you have within the cell the nucleus. They've pulled the nucleus out, and looking at the nucleus, then you have your nucleolus here. And then these X structures, which they're isolating one out, that's known as a chromosome. And if you uncoil the chromosome, you're going to start to see the chromatin, which is going to be the DNA material wrapping around proteins that are called histones. And if we continue to uncoil that, we'll eventually get to that double helix structure that we're familiar with for the DNA. And so within the nucleus, the DNA starts to coil around the proteins, making chromatin. Continue coiling, we'll get that chromosome structure, which again we find in the nucleus of the cell. The chromatin material is usually dispersed until mitosis. Now, mitosis is part of the cell division process, and our next unit will go through and talk about mitosis and the steps. But this is usually in the beginning of mitosis in prophase. This is usually when the chromatin is going to start to condense, coiling tighter and tighter so that we can see the chromosomes. As we take a look at DNA replication, let me get the pen, we need to look at the cell cycle. And this diagram is representing the cell cycle. The cell cycle, in other words, the whole life of the cell, we have a growth phase, which we'll call G1 phase, continuing through an S phase, which is known as synthesis. This is where DNA is replicated in the S or synthesis stage. And then we have our G2 phase, which is more growth, and then it's preparing for mitosis. So G1, S, and G2, those are known as the interphase. Interphase again is G1, S, and G2. This is where growth, DNA replication, and growth and preparing for mitosis occurs. For now, we're focusing on the S phase or synthesis phase where the DNA is replicated. As part of the cell cycle, the cell is going to divide to make more cells. But before the cell can divide, we need to ensure that the genetic information is passed on to the future cells. In order to do that, the DNA has to replicate first. If it doesn't replicate, then the new cells wouldn't have genetic information. So it's important that the DNA is replicated before cell division occurs. And we'll get into mitosis and cytokinesis in our next unit. Let me get my pen. Okay, each strand of the DNA double helix has all the needed information, and we need to be able to make a copy of that. So what happens when you have your DNA structure, that double helix, you'll have an enzyme that's going to come in and actually break apart the hydrogen bonds that were holding the nitrogen bases together. That enzyme's known as helicase. And then once helicase helps separate the double helix into individual strands, these are known as our parent strands, the parent being the original ones that we start with. Then you'll have another enzyme known as a primase that deposits primer. It puts primer down on that parent cell, so then a third enzyme can come in known as polymerase and help develop the new strands, the complementary pair. And then in the end, what you have are two identical strands of DNA, two double helixes that are identical to one another, and that is through the process of DNA replication. DNA replication, that literally means making a copy of the DNA, and that copy needs to be identical to each other, and then they have to be identical to the original strand. And these are the four enzymes that we're referencing. There are many more. We're just kind of simplifying this process for now. But again, you have your DNA. Helicase comes in and breaks the hydrogen bonds that are connecting those nitrogen bases so they can separate. This is showing it's separated. And then you'll have your primase comes in and it deposits a primer down on that parent strand, or the template as they're calling it here. Puts a primer down so that the polymerase knows what it needs to build for the complementary pair. And so your primase comes in and builds that new strand, or the complementary strand. And you'll notice that this complementary strand, okay, one is moving in this direction and one is moving in this direction. And that has to do with something known as 5' and 3', and we'll look at that in just a minute. Looking again at the DNA, again, you've got your helicase is going to help to unzips the DNA, separating it into two parent strands. And then you will have a primase that deposits a primer. And then polymerase is going to come in and make the new strand, depositing down the complementary pairs. So that in the end, we can have two identical DNA strands. The DNA polymerase, this is your DNA polymerase. It's another enzyme again. It's going to put a new nucleotide on the other side. It would bring, if you look at this nucleotide that's represented, it's going to put another nucleotide over here. I mean, we've got your nitrogen base, your sugar, and then your phosphate. It's building that new strand on the DNA. The polymerase is also responsible for proofreading. As it's building those new strands, the polymerase makes about 1 in 10,000 errors in the base pairing. Remember, adenine must go with thymine, guanine must go with cytosine. Then there are further enzymes that come through and they proofread it again, looking for mistakes. Once the enzymes proofread the polymerase, we're now looking at errors of 1 in 1 billion. All right, guys, remember when I said that on the diagram when I was showing you how the arrows moved in opposite directions? That's because DNA is anti-parallel, and you need to know this term, anti-parallel. The anti-parallel is the direction in which that new strand is made. On this particular structure that we're looking at for this segment of DNA, our new strand on the left would move down, and then this one on the right would move up. That's because the DNA polymerase moves from a 5' to a 3' direction. Again, from a 5' to a 3'. Now, understanding the 5' and 3', don't get too caught up in it. It has to do with numbering the sugar, the carbons within the sugar. And so at each one of these points on our sugar, there's a carbon. And the starting point is based on that first oxygen. And so if you'll look at your diagram, you'll notice our 3' is in the down position on the left, but on the right, it's on the top. Again, it has to do with numbering based on where the oxygen is located, where they start the numbering of carbons. You guys don't have to worry about that at this level, but what you do need to know is that the DNA polymerase, again, is going to move from a 5' to a 3', 5' to 3', and that's where we get the anti-parallel. One's going in one direction and the other goes in the opposite direction. Because it moves from a 5' to 3', one side is going to end up having fragments of our complementary pairs. And since there's fragments, we need to be able to connect those fragments on the DNA structure, and that's where ligase comes in. Ligase is going to glue those fragments together. DNA damage and repair. There are chemicals in ultraviolet radiation that damage the DNA in our body cells, and our body cells are known as somatic cells. Make sure y'all add that in your notes. Body cells and somatic cells are interchangeable because then we have sex cells that are different. The cells continuously need to be repaired. The DNA gets damaged and we have to repair that. The DNA polymerase and the DNA ligase replace and bond new nucleotides together in order to help keep this process going. Taking a look at DNA again for the replication, you have your 2 strands of DNA. Remember, helicase would come through, separate the strands. Then your primase would deposit a primer, allowing for the DNA polymerase to come in and make the new strand, which we call a daughter strand. Daughter strand just means the new one. New strand means daughter strand. And then the original strand that was there at the beginning, that's known as our parental strands. We have the parent strand and the daughter strands.